Hi, you are welcome to my YouTube channel. In today's video, we are going to look at the concept of molecular biology. And my name is Mukhtar Aliyu Ikara from the Department of Biochemistry, ABU area. Introduction to molecular biology. Molecular biology is the study of biological processes at the molecular level. So when you said biological processes, it includes metabolism, the functions of each and every cell of our body, such as cellular recognition, cellular adhesion, cellular respiration, digestion. All these are examples of biological processes. So in molecular biology, we are studying the biological processes using a molecule. So therefore, these molecules, of course, they are the ones that are responsible for the cellular differentiation and specialization. So if you see a particular cells differ from one another, of course, that was actually due to different proteins or different biomolecules that are produced by that specific types of cell. So molecular biology, it focuses on the interaction between various systems of the cells, including DNA, RNA, and proteins. So therefore, in molecular biology, it focuses on the interaction between various systems of a cell, including DNA, RNA, and proteins. So in molecular biology, we are using the interaction between DNA, RNA, and proteins, understand the different systems of a cell, including the structure and the functions of the cell. Like, for example, in molecular biology, these biomolecules, DNA and RNA, are strongly or they are related with one another. Like, for example, in this molecular biology, first we have a DNA in the nucleus. And then in this DNA, there are about 25,000 genes that are actually present in the DNA sequence. And each of that genes is responsible for producing a specific messenger RNA. And each of these specific messenger RNA is responsible for producing a specific types of proteins, which will actually lead to the structure and the functions of the cell that produce the protein. Like, for example, the DNA first is converted into RNA, and then the RNA is converted into protein. It's converted into protein. So first, DNA is converted into RNA. So a specific gene from the DNA will be transcribed through the process called transcription into RNA, and then the RNA will translate into a protein. And that protein is what actually determines the functions of each and every cells in our body. Like, for example, in the red blood cells, we have a protein called hemoglobin. And of course, that hemoglobin is produced because we have a specific genes that is responsible for producing the hemoglobin. So hemoglobin is what actually gives the red blood cell its function, as well as it is bioconcave shelf. So then the next thing after this, we should understand that we have the next thing actually is understanding the interaction. So understanding the interaction between these macromolecules have actually helped to explain how genes are regulated and expressed. So what is gene regulation and gene expression? So when you say gene regulation, it means how a specific DNA sequence is converted or is actually translated into a specific protein. So that is gene expression. So when a DNA is translated into a protein, that is what we describe as expression. And then regulation of the expression is actually to control at what time these proteins are supposed to be expressed and at what time are not expected the proteins to be expressed. So the regulation of this expression of the genes So what maintain our cellular structure and function that make us also live healthy. And whenever there is no control in gene expression, once there is no control in gene expression, it means that there will be a problem, there will be a disease. So like now, for example, in this gene expression, it is very, very understanding, or it is very, very important to understand how these proteins are expressed briefly. So generally, in a living cells, a living and a functional cells, the first molecules or RNA that is generated from the DNA is called messenger RNA. It's called messenger RNA. And of course, with the help of this messenger RNA, the proteins will be translated, because this messenger RNA, it's like contain a language that will be translated into a specific amino acid, and these amino acids will join together to produce a functional protein. But how? First, the messenger RNA will be actually, or is generally produced in the nucleus, and this messenger RNA will be transported to the cytoplasm. And in the cytoplasm, we have a transfer RNA, which is a carrier for a specific protein. So with the help of transfer RNA and the ribosome, this is a ribosome with both A side and P side. So with the help of ribosomes and transfer RNA, the messenger RNA will be translated into a protein. So the major functions of the transfer RNA is to carry a specific amino acid which will be recognized by the sequence, by the triplet codons in the messenger RNA. So this is actually a brief description of what this molecular biology is. So you have a DNA converted into RNA, and then the RNA will be converted into proteins. And the proteins is what determine the functions and the structure of cell. There's a gene for hemoglobin, and that is why the gene will be first transcribed into messenger RNA. Then the messenger RNA, with the help of transfer RNA and the ribosome in the cytoplasm, will be translated into proteins. So the proteins, which is hemoglobin at this point, it will be responsible for the structure and the functions of the red blood cell. And of course, as I explained, gene expression is when you have DNA converted to RNA and RNA to proteins. So to control how these genes are expressed and when are they supposed to express, we are going to see that as we continue with the lecture. So now, the next thing actually is the history of molecular biology. So the field emerged in the mid of 20th century with the discovery of the structure of the DNA. So the history of molecular biology is started with the discovery of the DNA by Mr. And of course, the key figures like James Watson, Francis Crick, and Rosalind Franklin have made a significant contributions in the field of molecular biology. And we are going to look at their contributions one after the other. And of course, the establishment of techniques such as DNA sequencing, it of course revolutionized the molecular biology research. Because when you say DNA sequence, it's actually to determine the sequence, the number and the sequence of arrangement of the of the nucleotide sequences that are found in the DNA. Like, for example, the DNA sequencing after actually Human Genome Project, it was revealed that the human genome contains about 3 billion base pair, 3 billion nucleotides, 3 billion base pairs. It's a huge number. So scientists was able to discover that after the complete Human Genome Project, the whole genome of human have been sequenced, and of course, it revealed that there are about 3 billion nucleotide sequence. And of course, one of the very interesting things that we should understand at this point is, of course, we are going to look at the contribution made by each of the scientists. So the history of molecular biology, of course, has started with the Mendel in the year 1865. So Johann Gregor Mendel was the first scientist that was able to document patterns of heredity in key plants. That is how characters transmit from generation to generation. So Johann Gregor Mendel is the first scientist that was able to demonstrate that in the year 1865, followed by Mr. So Mr. is also the first scientist that was able to identify DNA. He was able to identify DNA, and at that time he named the DNA nucleic. So these are actually the first scientists that actually tried to unravel the mystery of biomolecules in a cell. So Mendel documented the patterns of heredity in people, and that is how characters pass across generations, followed by the Mixture, who was actually the first scientist that was able to identify the DNA, which he actually named it nuclein. And then after that, then the next thing is, of course, Sutton and Boveri. So Sutton and Boveri in the year 1902, they are the first scientists that proposed the chromosome theory of heredity. That is how chromosomes participate in heredity, that is transfer of characters across generation. And then after that, Morgan and his, of course, fly room, that is colleagues, confirm that the chromosomes theory of heredity, that the chromosomes is very, very essential in actually transmitting characters from parents to their offspring. Later on, the scientist Muller in the year 1927 was able to demonstrate or shows that the X-ray can induce mutations. And when we said a mutations is, of course, a permanent change in a nucleotide sequence, which can have positive and the negative effect in the organisms that actually induce the mutations. So this Muller was able to establish that X-rays can induce mutations in the genome. And then the next thing after Muller is, of course, Griffiths. The Griffiths was actually be able to demonstrate that in the 1928, in the transformation experiment that transformed non-pathogenic bacteria strains to pathogenic. So he's the first scientist that actually established a transformation experiment where you can transform non-pathogenic bacteria strain into autopathogenic. That is, when you have non-pathogenic bacteria, then through mutations and some genetics rearrangement, you can actually change that non-pathogenic bacteria strain to pathogenic. And that is why there is issue of bioterrorism, where you can use a bacteria or you can use microorganisms or any product from the microorganisms for terrorism. And then the next thing after this Griffiths is McClinton. So McClinton is the first scientist that was able to demonstrate genetic recombination in corn. So he demonstrated genetic recombination in corn. So these are actually some of the discovery of these scientists that actually plays a significant role in the journey of molecular biology. And then the next one after Max Clinton is Biddle and Tatun in the year 1941. This scientist was able to describe that one gene, one enzyme hypothesis, that one gene is responsible for producing a specific protein or enzymes. As I explained earlier, there are like approximately 25,000 genes in humans, and each of these genes is responsible for producing a specific protein. That is one gene, one protein, or one gene, one enzyme. That is Biddle and Tartan experiment in the year 1941. So it means that let's actually make this calculation. If human being have approximately 25,000 genes, means that humans also have approximately 25,000 proteins. Then the next thing after this Biddle and Tatwin is also another series of scientists, which is Avery, MacLeod, and Mackey. They are the first scientists that show that DNA is actually transforming a principle that is responsible for heredity, that the DNA is responsible for transmitting a trait from parent to their offspring. It means that the character that we inherit from our parent is through the DNA. So that is why sometimes I call DNA as a language of life, because DNA is what determines you. DNA is what actually that actually give you all the features and the characteristics that you have. That is why, because there is the issue of genetics recombination by McClinton, because during sexual intercourse, when fertilization takes place, it means that you are going to have the combination, or the zygote develop will have the combined DNA of father and mother. And that is why our child usually have the character of both mother and mother, that is maternal and paternal trait, because of the recombined DNA that was actually generated from their parent during meiosis. And then the next thing is actually Chagabs in the year 1950. So Chagabs was actually the first scientist that discovers that a nucleotide pair with one another, that adenine pair with thymine in the DNA, and cytosine pair with guanine. And of course, the number of adenine in a particular human DNA or in a particular DNA reflect exactly the number of thymine, and the number of cytosine reflects exactly the number of guanine. So that is Chagab's rules. He tried to identify how one nucleotide pair with one another in the double helix of DNA. And of course, between adenine and thymine, they actually pair by two hydrogen bond, and between cytosine and guanine, they pair through three hydrogen bonding. And then the next thing is actually Hale or Hashi chess. Hashi chess in the year Hashi chess in the year um in the year 1952, they actually use radioactive labeling to prove that DNA is responsible for heredity. So these are the scientists that clearly demonstrate that DNA is responsible for heredity using radioactive labeling. And then the next one is, of course, Watson and Crick. Watson and Crick in the year 1953, they are the scientists that proposed the double helix structure of DNA. And then after that, then we have Jacob and Monod, which are actually scientists that propose the existence of mRNA. So these are the scientists that actually make the contribution. And of course, in 1990s, a series of scientists was able to actually complete human genome at different labs across 6 different countries. So the actually project takes about 13 years to be completed. So this is actually the journey of molecular biology, and this is how it actually started with the series of experiment, with the series of discovery by different scientists across the world by that time. So then the next thing, of course, after this is the central dogma of molecular biology. So as I explained, that the central dogma is describe the flow of genetics information in biological system. So central dogma describe the flow of genetics information in biological system. So let's see how this genetics uh, information flow in the biological system. So first, the central dogma is trying to explain how DNA is transcribed into RNA, which is then translated into protein. So in central dogma, the DNA is transcribed into RNA through transcription, and then the RNA is converted into proteins through a process called translation. So this process is crucial for cell function and inheritance of traits. Like for example, if in a particular cells, a proteins that is produced, let's say, is insulin, so that cells will actually be responsible for the control of the blood glucose level because of the insulin it produces. Although hormones, also insulin is a hormones, so sometimes hormones do not actually elicit their functions at exactly where they are produced. A liver is involved in detoxification of toxic substances. Toxic substances due to proteins that are generated from there. So this central dogma of molecular biology is important in cellular functions and the inheritance of traits. So this is how the central dogma work. First, the DNA polymerase, an enzyme called DNA polymerase is converting, or it converts DNA to DNA, and the process is called transcription. Sorry, it's called replication. So when DNA is converted itself to another molecule of DNA, the process is called replication, and the major and the key enzymes that is responsible for doing that is DNA polymerase. And then the DNA polymerase also, then of course the DNA produced can be converted into RNA, and the process is called transcription, and the major enzymes is RNA polymerase. And in the case of this RNA, it can also be converted into protein by the ribosome by the process called translation. So this is actually a brief description of central dogma of molecular biology. So then next thing is DNA structure. So the DNA is composed of two strands forming a double helix, and each strand consists of nucleotides. So each of these strands, it consists of a nucleotide which actually joined together by glycosidic bonds. And of course, one nucleotide joined with another nucleotide by phosphodiester bond. So each of these nucleotides is actually made up of different components. So nucleotide is made up of a sugar, phosphate group, and nitrogenous base. So the sequence of these bases encode genetics information essential for life. So this sequence, that is nucleotide, are responsible for encoding the genetics information that is essential for life, that is protein. So let's look at this nucleotide. So nucleotide is made up of phosphate group, sugar, is pentose sugar, we call it, of course, ribose sugar. So in DNA is called ribose sugar, while in DNA is called deoxyribose sugar, while this in RNA is called ribose sugar. So it is a 5-carbon sugar. And then the next thing is bases. So in DNA, we have 4 bases. A stand for adenine, T stand for thymine, C stand for cytosine, and then G stand for guanine. So these are the nucleotides or nitrogenous bases that are found in the nucleotide of DNA. And then of course, these nucleotides form phospho or sugar phosphate backbone, which actually leads to the pairing of another sequence in order to produce a complete double-stranded DNA. So of course, this is the double-stranded DNA. So this is actually a brief description of DNA. It's made up of sugar, phosphate group, and nitrogenous bases. And all the nitrogenous bases, the phosphate group, and the sugar are described in the image. And then in each of the strands of these nucleotide bases pair with one another to form a complete double helix structure of DNA. So the next thing that we are going to look at is different types of RNA and their function. So generally, we have so many different types of RNA. We have different types of RNA. There are more than 10. But of course, in this video, we are going to look at 3. So the RNA plays several roles in a cell, including messenger RNA, ribosomal RNA, and transfer RNA. So in molecular biology, these RNA, they are very, very important in producing or expressing the functional protein. So the major function of messenger RNA, it actually carries genetics information from DNA to ribosome for protein synthesis. So RNA is what actually carries the genetics information. And then the next one is transfer RNA. So transfer RNA brings amino acid to ribosome, facilitating the translation process. So this is how it is. We have here, this is the messenger RNA. So this is actually the messenger RNA, and this is the ribosome, and this is the transfer RNA. Of course, this is the loaded amino acid. So messenger RNA, ribosomal RNA, as well as transfer RNA combined together to make sure that proteins is actually successfully expressed, that is produced by the cell. So then the next thing is protein synthesis. So how proteins are synthesized? So protein synthesis involves transcription and translation processes. So during transcription, mRNA is synthesized from a DNA template in the nucleus, and then of course the translation occurs in the cytoplasm, where the ribosome synthesizes proteins from mRNA structures. So first, mRNA is synthesized in a nucleus using a DNA as a template, and then the messenger RNA will be transported to the cytoplasm, where ribosomes, that is the ribosomal RNA and transfer RNA, combined together with the messenger RNA to make sure that there is an effective protein synthesis. So let's look at it briefly, actually. So you see, first, we have, this is the DNA, and then the DNA is converted into messenger RNA. So the process is called transcription, and of course, you can see clearly this process is taking place in the nucleus. And then, of course, from the nucleus, the messenger RNA here, look at it, is transported to the cytoplasm. So, of course, in the cytoplasm, the messenger RNA, of course, with the transfer RNA, joined together to produce the proteins through the process called translation. So you see, this is, of course, an amino acids that are linked to one another and ribosomes to form the protein encoded by the messenger RNA. So this is actually, all this lecture that we are doing is brief. So as time goes on, we are going to have a detailed explanation of how these proteins are fully synthesized, how RNA replication and transcription take place. So the next thing is actually gene regulation. So a gene regulation is a process by which cells control gene expression levels. So gene regulation, as we said, when DNA is converted into proteins, that is gene expression. So gene regulation is actually the process by which cells in the body will control gene expression levels. They will actually control the up, down, on and off regulation of gene at a particular time how much or how many of that proteins will be produced and how many will be reduced. And at some point, it will completely off. That is, the expression will stop, or at some point, the expression will be actually activated. So this is actually gene expression briefly. And then this gene regulation, it involves various mechanisms, including transcription factors is a factors that also influence the expression of the gene, as well as the epigenetics modification. So transcription factors are factors that always influence or that help in actually translating or sorry transcribing DNA into messenger RNA. So these transcription factors are very important in helping the RNA polymerase which converts DNA into RNA. And there's also genetics modification, which is actually a modification system that actually control the expression of the genes without affecting the integrity of the DNA molecule. And then the next thing, so proper regulation is essential for development, differentiation, and response to environmental signal. So if the genes are properly regulated, so it is actually essential for development, differentiation, and response to different signals. And of course, sometimes it's due to improper gene regulation. That is why we have some of our child are coming off with some genetic disorder. So this is actually there are different points where you can control this gene regulation. So there are various points of gene regulation, starting from the signal that will be sent to the cell. It is the amount of the signals also affect how many of the genes product will be produced. So after the signal, then there is a chromosome. So the chromosomes involve the chromatin involves so many modification system, which is called epigenetics modification. So there's a chromatin modification, which is also uh, another way, or which is similar to epigenetics modification, which includes DNA unpacking, which involve histone acetylation and DNA deamination. So histone acetylation and DNA demethylation are actually examples of epigenetics modifications that are used to unpack the DNA, because generally DNA and chromatin or an histone protein are tightly packed together, and that is why we have chromosome. So in some cases, these DNA and proteins, they are tightly packed. So to unwind them for the transcription and translation to take place, it used to be very, very, very difficult. So of course, uh, DNA unpacking, it is very, very important to have to expose the DNA for transcription and translation to take place. And at the same time, the DNA will be converted to RNA at actually to RNA at transcription level. So what I want us to understand, we can control gene regulation from the signal point of view, from chromatin modification point of view, or also transcription point of view. So gene available for transcription, that is why the DNA is converted into RNA that is actually specific to the transcription factor or the gene that is available after epigenetics modification. And then after transcription, we have a primary transcript that is, of course, immature RNA. So the immature RNA, which is the primary transcript, will now be processed. So in the primary transcript, we have both introns and exon. So exon is actually a coding region of a gene, while intron is a non-coding. So therefore, the non-coding region will be actually removed through a spliceosome or spliceosome complex activation system. So all the introns is like a molecular scissors where all the introns will be cut out, and at the end, and of course, this RNA processing involves so many things apart from the removal of introns. Then there is also post-transcriptional modification, which involve addition of HAP to the matured RNA, tailing, that is to add tau. So the HAP is actually 7-methyl guanosine, while the tail is poly A tail. So after the processing, what the cap, the tail, and of course the removal of the introns will take place. So after that, then the transcription, after the transcript will be actually be moving or will be transported from the nucleus to the cytoplasm. So here we have, of course, we can see clearly, here we have mRNA in the cytoplasm, which actually in some cases it will be degraded if it is not needed, and if it is needed, then it will actually be translated into a polypeptide. So this polypeptide, of course, it will undergo post-translational modification. So the cleavage of chemical modification, transport to proteins, and destination are taking place at the same time under post-transcriptional modification. So after that post-translational modification, you'll have an active protein. So degradation of this protein will take place after the proteins actually elicit its function. So once proteins elicit its function, so it is no longer needed. So it will be degraded and actually converted into individual amino acid, which will be used by other transport RNA to synthesize another protein. So ladies and gentlemen, let's meet in part 2. This is just a brief introduction in molecular biology. I hope you enjoy the lecture. Please don't forget to subscribe and join the YouTube community. Thank you very much.